Good morning, everybody. Welcome to the first MPG primer of the season. I am thrilled to introduce Dan Snelling. He's a postdoc in the Walsh lab at Boston Children's Hospital. He received his PhD at Duke University working with Douglas Marchuk, studying how somatic mutations drive the formation and progression of vascular malformations. Currently, he's in the Walsh lab, and he's developing methods for tracking cell lineage using somatic mutations and applying them to understand the impact of somatic mutations, somatic mosaicism in neurofibromatosis. But that's actually not what he's talking about today at all. He's talking about the awesome power of genetic linkage. And this is something that comes up often with trainees on trying to understand LD. It's extremely important, and we're excited to have him. Please ask questions throughout. There's microphones in the center, and we'll also have time at the end for questions. Thank you, Dan.
Great. All right, thanks so much, Michelle, for the introduction and for the invitation to give this talk. It's my pleasure to give a more basic talk today that's not so much about my specific science, though there might be a little bit about that at the end, and more about the basic concept of genetic linkage and linkage disequilibrium and phasing, and how this is one of the most important tools that we use as geneticists. And so I figured today that I would take more of a historical perspective on linkage to kind of understand how we got to where we are, and hopefully in the process, learn a little bit about linkage, LD, and phasing. And so naturally, when starting a discussion about linkage, the best place to start is by talking about hair, and specifically my hair, and how I'm blessed to have a full head of it, because looking at my family tree, it's not really clear how long that's going to be the case. And so male pattern baldness has long been known to have a very strong hereditary component, but the mechanism of inheritance had not been known for a long time. Some really early studies had suggested that it was autosomal dominant in males and autosomal recessive in females, while other studies and common folklore had suggested it was actually an X-linked trait, and so came from the mother's side of the family. And so in 2005, Axel Hilmar and his colleagues set out to actually find the location of the gene that causes male pattern baldness. And so they knew that the gene, or at least they suspected that the gene was on the X chromosome, but they didn't know exactly where it was. And so they performed a really classic linkage study where selecting just a group of 95 families and genotyping just a few dozen markers on the X chromosome, they were able to narrow the location of the gene causing male pattern baldness to a window on the P arm of chromosome X. And so here at the bottom, you can see the different markers that they genotyped in the assay. And note that this was before we had things like SNP chips and next generation sequencing. So all of this was done manually by, I'm sure, graduate students and postdocs and undergraduates. And you can see this LOD score increase on an arm of the chromosome. And this actually corresponds to the location of the androgen receptor, which is responsible for binding testosterone in males, and it seemed like a very likely candidate at the time. And so today, with modern GWAS, we have much larger cohort sizes, and we genotype hundreds of thousands or millions of variants within a single assay. And we do find that there's a lot of other factors across the genome that contribute to male pattern baldness, but that initial finding on the X chromosome remains our strongest hit to date. And so I bring this up as an example of how we can use linkage to answer one of the most challenging problems in genetics. That is, how do we actually find the variants driving a phenotype? Whether that phenotype be baldness or disease, or even just what makes us human. And so to take a historical perspective on linkage, we need to start by talking about Mendel, who worked on peas from 1856 to 1863. And he was really the first to realize that traits such as the color of peas and the shape of peas and their seeds were heritable units that followed a regular pattern that could be reliably predicted from the phenotypes of the parents. So here crossing yellow seeds that are smooth and green seeds that are wrinkled, they were able to show, or rather he was able to show, this very regular pattern of inheritance where you would get seeds with all mixtures of these phenotypes in this very stereotyped pattern of 9-3-3-1. And so this observation eventually led to the law of independent assortment, which is that two traits are inherited independently of one another. And so whether you inherit one trait is totally independent of whether or not you inherit a second trait. And so the first problem with this idea really came out of the rediscovery of Mendel's work in the early 1900s, when a group with Bateson, Saunders, and Punnett were trying to reproduce some of Mendel's findings. And so they were studying flowers that had purple flowers and long pollen, and red flowers with round pollen. And so by crossing the F1s of that cross, they expected to see that same 9-3-3-1 ratio that Mendel had described. But in fact, what they observed was a stark difference from that 9-3-3-1 ratio. And you can see here that there are very few flowers that they obtained that have purple flowers and round pollen, or red flowers and long pollen. And at the time, they didn't really have a great explanation for why that was. And it wasn't really until a decade later when Hunt Morgan, who at the time had been working on similarly linked traits in the sex chromosomes of Drosophila, and his student Sturtevant, who had the insight that by looking at variance in the strength of linkage between two traits, you could actually arrange the gene causing those traits on a linear entity, which we now know as a chromosome, and two traits that were more strongly linked were more close together on that linear entity, and traits that were less linked were further apart. And this was really the first genetic map that really launched the field of family-based linkage studies in human genetics. And so Sturtevant also noted that in cases where you break this linkage, so in the previous example where we have purple flowers with round pollen, these are cases where we've had a crossing over event on a chromosome that actually occurred in between the site of the genes causing those two traits. And so this was really beautifully demonstrated later in the 30s by Barbara McClintock before her Nobel-winning work on transposons, and her colleague Harriet Crichton, who at the time had been developing cytogenetic tools in maize. And so they were studying traits in corn, maize, that were similarly linked. And so here looking at colored and colorless kernels and smooth and wrinkled seeds, by performing a cross between hybrid, that is heterozygous for both of these traits, and the parent that is homozygous for the recessive allele, you would expect to find an even distribution of phenotypes for all of these sets. And in fact, similar to what Bateson, Saunders, and Punnett found, they see a deviation from that expectation, and that there's very few offspring that have a recombination event between those two sites. And this is really reflecting that those two, the genes driving those two traits are very close on chromosome 9 in maize. And so McClintock and Crichton were studying various different pairs of phenotypes within maize. And one had a particularly striking characteristic that enabled them to really see linkage with their own eyes. And that's this pair of studying kernel color and waxy endosperm. And so the characteristic that really enabled this is that when they looked at the chromosomes driving these traits, they found that using these cytogenetic tools, they could actually visually distinguish one chromosome from the other. And that was because the chromosome harboring the colored allele and the waxy allele had a visible knob at the end, this being heterochromatin, a very dense area of heterochromatin. And it also had an extra piece at the end. And so they had these markers on either end of the chromosome that they could use a microscope to actually visually observe. And so when they did this cross, what they found, similarly to what they had here, they had some number of offspring that had a recombination between those events, less than would be expected by independent assortment. And when they looked at the chromosomes of those offspring, they could see that all of those recombinants actually had the knob at one end and missing the extra piece at the other, or the extra piece at one end missing the knob at the other. So actually visually saw the recombination event happen, or could at least infer that it must have happened. And so this series of observations with Sturtevant and genetic maps and the idea that genes are on this physical linear entity and linkage is the phenomenon resulting from crossing over during meiosis, really led to the field of family-based linkage analysis that has been probably the most powerful tool for understanding the cause of Mendelian disease. And so here in this example, we're looking at one pair of chromosomes from an initial coupling of an affected male and an unaffected female. And throughout the generations, we can see that children are affected as well. And what we can do in family-based linkage is just genotype a handful of markers across the chromosome. And these markers are broken up over generation by recombination events. And so what we see if we, after doing all of this analysis and looking at all of the identified markers in the affected and unaffected individuals, we can see that there's a region of the chromosome that harbors this C allele in all of the affected progeny and does not harbor that C allele in all of the unaffected progeny. And so this C allele may not be the actual cause of the disease, but is something nearby such that we are observing linkage between the actual genic cause of disease and the C allele that we were able to genotype. And so this had been hugely successful for studying Mendelian diseases, and really high-profile successes like mapping the gene for cystic fibrosis and Huntington's. And over the years, there have been thousands more Mendelian traits that have been mapped to causal genes using family-based linkage analysis. However, it really was not so successful for studying complex disease. And so here in pink, you can see the meteoric success of linkage for studying Mendelian traits throughout the 80s and 90s, but relatively little progress for understanding complex disease, which was, I think, particularly disappointing at the time, because there was a lot more effort being put into understanding these very common complex diseases than there was effort understanding these rare but very numerous Mendelian diseases. And so the reason this is just a mathematical consequence of the way effect sizes and penetrance works, where in Mendelian disease, you can be quite confident, at least for very high penetrance variants, that if you have the genotype, you will have the phenotype. And if you do not have the genotype, there is no instance in which you have the phenotype. So very high penetrance, very high effect sizes. And this is not true for complex diseases. I think we all know there are usually many genetic factors that contribute to complex disease. And there's also many non-genetic factors that also contribute to genetic disease. And so what are we to do? So in the light of all of this work trying to use linkage, with few exceptions, unsuccessfully in complex disease, Neil Risch and Kathleen Marikangas were trying to get a mathematical handle on this difference. And they modeled how many families you might need to observe an effect in a variant that wasn't such a high effect size. And so here, looking at a very popular design for linkage studies at the time, where you look at affected siblings and find areas of the genome that are shared between those siblings, If we look at effect sizes of 1.5 to 4 times more likely to have the disease, it turns out that as demonstrated by the lack of success in linkage studies, you really need an astronomical number of families to detect variants at small effect sizes using linkage-based analysis. But what was really heartening is that they proposed this alternative study design where instead of trying to map the gene using linkage, you instead test for direct association between some putatively causal variant and whether or not that person inherited the disease. And these numbers were, as you can see, much more favorable, even for very low effect sizes and for very rare population allele frequencies. And one other benefit of this is that you didn't need to capture these massive families, but rather you could focus just on affected individuals and their parents for doing these kinds of association studies. And so it was much more accessible than collecting very large pedigrees. However, the main limitation of association is that you need to actually know where to look. And so if you're coming into a complex disease and you don't have any prior of where that causal gene may be, and even what variants specifically are driving the phenotype, it's really hard to carry out an association study, especially in these days when, we don't even really fully understand the degree of variation that's present within different human populations. And we really don't have the tools to genotype variants across the genome at any reasonable level of throughput. And so at this time, we kind of understood that association seemed to be the way forward for understanding complex disease. But what do we actually need to make those kinds of studies a reality? And so one thing that had been sorely missing was, as I said, just an understanding of genetic variation within the human genome. And so this was really helped along by several large projects to sequence or at least genotype individuals from many different populations and get an idea of how much variation is there in the human genome. And this ultimately culminated in many of the databases that we use all the time today, like dbSNP and Nomad and many others that have cropped up over the years. And so one of the key insights from these basic studies of genetic variation in humans has been that there's a lot of variation in humans. Each individual on average has about 4 million variants. And as expected from the out of Africa hypothesis, African populations typically have far more at about 5 million variants. But what was really important to recognize is that the vast majority of these variants are actually shared between populations. They're very old. And so in this graph, the colored bars are noting variants that are unique to that population or to that individual, whereas the two gray bars are variants that are shared throughout the entire world or just within that continent. And what you'll notice is that the vast majority in most populations, the vast majority of variants are shared across continents or even across the world. And this makes the problem a little bit more tractable, where everyone has 4 million variants, but in total, there's only about 10 million common variants in humans. And the other key observation that kind of follows from this is that most variants are really old. And this is the core concept of linkage disequilibrium, where generally, in most cases, a variant that we observe in an individual happened one time in some ancestor and is now present in many people as a consequence of heredity, rather than that mutation occurring many different times. And so because the mutations are old and occurred once, these variants are only separated from their surrounding sequence by recombination events happening across generations. And so here we can see in present-day chromosomes, if you look in the area around some marker of interest, they actually all share sequence identity with that ancestral chromosome. And this is, again, just another manifestation of the property of linkage, where the closer you are to a marker and the more recent that mutation is, the more likely you are to have the same sequence surrounding it. And so one of the important properties of LD is that it erodes over generations. So LD is a function of meiotic recombination events, separating out those alleles and kind of making that haplotype smaller over time. And so over a few generations, you can see there's some recombinations that make these blocks of color smaller as you go. But then over hundreds and thousands of generations, these blocks erode away into very small chunks of DNA. But note that this is a very long process. And for most of the variants we look at today, we can see a reasonable level of linkage disequilibrium that we can leverage in many of our genetic studies. And so another key observation from these basic studies of genetic variation in humans is this observation that recombination is not random across the genome. There are hotspots of recombination. And as a consequence of that, we get this phenomenon of haplotype blocks, where there are sets of SNPs or variants along the genome that are almost never broken up by recombination. And so these are a really critical observation that led to eventually the HapMap project and mapping out haplotype blocks in humans and leveraging that information to very intelligently design genotyping arrays and actually perform association studies. But the other critical need for actually doing an association was really a technological innovation of being able to genotype many variants in parallel. And this really came around circa 2005 when the first high throughput genotyping arrays came out from Illumina and Affymetrix, where we're able to assay hundreds of thousands of variants at the same time in a single assay. And so the question that arises from this is, okay, we've got 10 million common variants within the human genome and room for, at the time, maybe 100,000 or a few 100,000 variants to fit on these genotyping arrays. So, if we're going to do association studies and expand this out to many hundreds and thousands of individuals per study, we need to have a really efficient way of assaying all of the genetic variation in humans without breaking the bank. And so how do we actually go about choosing SNPs to effectively design arrays for genotyping association studies? And the critical observation that makes this possible is that we can use LD to infer genotypes that we didn't actually directly assay in our arrays. And so that is if we have a haplotype block and a few variants in LD, and we look within the population and we see AAG, ACC, TAC, then if we design a genotyping array that only assays these first two variants, we can actually infer and in fact computationally fill in the third variant without ever needing to assay it directly, because we know that this variant is in tight LD with the other two, and therefore we don't actually need to assay it directly. And so through this principle, the HapMap project set out to actually map common haplotypes within humans and figure out how we can effectively genotype a small number of variants and get the maximal genotyping information from that via this idea. And so this is some information from the HapMap project looking at effectively a European population. And this idea of using a SNP that you actually assay to then infer other genotypes, that assayed SNP is the tag SNP. And when we look at power versus the number of markers we assay, we can see that by using the HapMap project and actually mapping out all of the haplotypes and common haplotypes in humans, we can effectively design tag SNPs that outperform arrays made by random SNPs by a factor of maybe 2 or 5-fold. And one thing that is really clear here is that in a hypothetical world where we didn't have LD, we would need to assay an order of magnitude more variants in order to get the same level of power that we would using SNPs designed with LD in mind. And so at the end of the day, what they find is that in order to get a pretty reasonable power for doing association studies, we can get by just assaying around 300,000 SNPs. And so this led to an explosion of GWAS studies and understanding in complex disease that I think ultimately is the best for another primer at another day, specifically dedicated to GWAS. But just making note that this has been a very successful strategy for identifying genic factors that drive complex disease. And so beyond just imputing SNPs using tag SNPs, one other really important property that we get from LD is the ability to actually impute haplotype structure, which is very important for many assays that we perform. And this is basically the idea that we have a list of variants across a chromosome, but how are they actually arranged on the maternal and paternal copies of the chromosome? And so by taking data from HapMap and 1,000 Human Genomes and other sources, we can take a population reference of how haplotypes are arranged and use that information to computationally infer how variants are aligned across a chromosome. And so this is, again, just leveraging the idea that if we have two variants that are close together and observed to be in very high LD, we can infer that they're present on the same haplotype. And so these days, there are many excellent tools for doing haplotype estimation and phasing, and here are some of the ones that I have used. But one thing to keep in mind when imputing phase from population data sets is that it is prone to error. And so this is an example where we have the true phase laid out in orange and blue. And when deriving phase from population data sets, often what you get is these phase switch errors, where in the individual there has been a recombination event relative to the population references that you're looking at. And so you end up having this switch in phase along the chromosome. And this can, of course, happen multiple times going down the chromosome. And so there's a few ways to ameliorate these errors. And one of them is or simply just using a bigger reference data set. And as time goes on and we sequence more and more individuals, this gets easier and easier as we've got massive data sets of individuals that we can use for doing really effective phase imputation from population data sets. And this is nice because it's easy to do. You don't need to actually do any experiments. You can just computationally get a really accurate phase estimation from any set of genotyping, whether that be whole genome sequencing or genotyping arrays. The other way is actually doing an experiment and using read-backed phasing to define haplotypes. And so the most useful tools for this have really been long-read sequencing and third-generation sequencing, where you can get reads that span kilobases or megabases and actually physically observe the phase across multiple variants over the genome. And the third, maybe the gold standard, is to get a trio and get DNA from mom and DNA from dad. And from that, in most cases, you can unambiguously derive the haplotypes present within the child. And so now in the second half of the study, I thought that I would talk a little bit about my own work and how I've used phasing to understand vascular malformations during my graduate work. And so when I was a graduate student at Duke, I was studying this rare Mendelian disease, hereditary hemorrhagic telangiectasia. And this is a disease of autosomal dominant caused by a mutation in 3 different genes involved in the same pathway. And it causes vascular malformations, the most prominent of them being mucosal telangiectasia. So these are like small red spots, as you can see, on mucosal membranes. So tongue, lips, eyeballs, fingers. But then more perniciously, they also cause arteriovenous malformations in internal organs. So the most prominent being lungs and brain. And you can see this mass of vasculature in the lung and in the brain that ultimately what's going on is in a normal artery vein connection, you have the major artery and veins separated by a capillary bed that in a very careful and controlled way allows blood to flow and nutrients to reach tissues in that area. However, in HHT, what happens is you get enlargement of those capillaries and effectively direct shunting between arterial and venous blood, which reduces the efficiency of things like respiration. And so you end up most often getting high output heart failure. And so when studying HHT, one observation that really drove our thinking was this disjointed nature of the genotype and the phenotype, where this is an autosomal dominant disease, so they have a heterozygous causal mutation in one of these genes. And the genotype, that causal mutation, is present in every cell in the body, but the phenotype, these vascular malformations, are inherently focal phenomenon. We get spots, we get discrete AVMs. And so we really wanted to understand what was underlying that difference. Why weren't we getting this kind of systemic vascular malformation that you might expect from having the mutation present in every cell? And so one thing that we thought could be the cause is a somatic mutation, similar to what you get in cancers. This is a mutation that arises post-zygotically in cells during development, and in fact, as we age. And these mutations are not present in every cell of the body and can be present very focally within multiple tissues or even within a single tissue. And so the thinking being that these vascular malformations are the result of a somatic mutation in a genetic Hudsonian two-hit mechanism. So this is that we inherit one null copy of the allele from the parents, and then a somatic mutation leads to loss of function in the same gene on the other functional copy. And that this event, following by likely many other non-genetic factors, is what's actually driving telangiectasia and vascular malformation. And so when setting out on the study to see if somatic mutations were actually the driver of vascular malformations in HHT, there were three things that we were expecting to find if this two-hit mechanism was indeed true. And so the first of those is that the mutations we identify would be in the same gene. So we've got a germline inherited mutation and a post-zygotic somatic mutation. And these are the three genes that are known to cause HHT. The expectation would be that the somatic mutation and the germline mutation occur in that same gene. The second would be that these mutations are all causing loss of function. So think frame shift, splice mutations. And then the third, and importantly, I think for demonstrating the benefits of phasing, is that the mutations are biallelic. And so if we have a loss of function mutation on one allele, and then later somatically we get another loss of function mutation, but in that same allele, we still have one functional copy of the gene and wouldn't expect vascular malformations to form. And so following from this hypothesis, we're expecting that the variants would be in trans, affecting both alleles such that no functional protein remains. And so in doing this, we set out with 20 samples of telangiectasia from individuals with HHT and used target capture to sequence really just a handful of genes that we suspected to be involved in vascular malformations from other studies at the time in other diseases, and then sequenced them to very high depth to identify somatic mutation. And then following that, do some validation with just amplicon sequencing and make sure that the variants we're identifying aren't artifactual. And indeed, what we found is that in 9 of those 20 somatic mutations, here showing the germline mutation, we also found a loss of function, or most likely to be loss of function mutation, in the same gene in each case. And of these, most of them were very obviously loss of function mutations. 5 of them were frame shift. 3 of them were in-frame indels affecting multiple amino acids. And the third was a splice site mutation affecting 4 bases after the exon-intron junction, which is a little far out. So we did, I don't show the data here, but we did experimentally validate that using splicing assays and showed that it did disrupt splicing and there were no full-length transcripts of the gene remaining. But one thing you'll note that is particularly challenging for studies in this nature is that unlike in cancer, where you basically get a clonal mass of cells that have expanded, in vascular malformations, it's not so clonal. And these somatic mutations are present at very low allele frequencies, which makes identifying them a little bit more challenging. And so really, for that third point of testing whether or not those mutations were in fact biallelic, we looked for read-back phasing, in this case, not using statistical phasing because these are rare mutations not present within population data sets. And so in order to get any information about phase, we need to actually observe phase. And so here's an example where we have the causal germline mutation on the right-hand side and a somatic mutation, a one base pair deletion causing a frame shift on the left-hand side. And when we look at all of the reads with the somatic mutation, you can observe that we have the wild type or the reference allele at the site of the germline mutation, indicating that these variants are in fact in trans and presumably in the cells with the mutation have no functional protein remaining. And we got lucky in this case where the germline and somatic mutation just happened to be very close together and could be phased directly from the reads used for discovery. In most cases, that wasn't true, and we turned to long read sequencing to figure out how those variants were arranged. And so here, an example on the bottom where we can see the discovery reads that these are very far apart and basically have no chance of directly phasing them from Illumina reads. But then with PacBio, we can very clearly see that all of the reads with the somatic mutation have the reference allele at the site of the germline mutation. And we were able to do this for 7 of the 9 variant pairs we identified, and in each case, they proved to be biallelic. And so the question then is, what about the other 11 malformations that we didn't find a mutation in? Is there some other mechanism by which these malformations can form, or is there something that we're missing? And our expectation is really that there's something that we're missing. For discovering the variants, we use targeted short read sequencing of just 16 genes, which is really great for picking up small events like single nucleotide variants and small indels, but much less useful for picking up very large variants that are just as likely to cause loss of function. And so to get at this question, we looked at traditional methods for identifying structural variation within the human genome. And one of the most common is using microarrays, so genotyping arrays similarly to what's used for GWAS, to look at SNPs across the chromosome. And here, looking at chromosome 21 in a normal individual, here we're plotting the alternate allele frequency. And you can see we've got a bunch of heterozygous variants, and we've got a bunch of homozygous variants for either the ref or alt allele. And then we have the LRR, which you can interpret as the genotyping intensity on the microarray. So an LRR of 1 is what we would expect to find for a normal diploid individual. LRR above 1 indicates the presence of a gain, and LRR below 1 would indicate the presence of a loss. And here in an example of trisomy 21, a patient with Down syndrome, you can see that the B allele frequency bifurcates, where you have one allele that rises in frequency, the one that is duplicated, and the other is decreased in its relative frequency. And similarly with the LRR, you can see that to be elevated above 1, indicating the presence of a chromosomal gain. And so it turns out that picking up variants with microarrays via traditional methods is very challenging when your allele frequency is extremely low. This is a germline event, so it's present in every cell in the body. But if we look at a somatic event that is only present in a handful of cells, we use these same metrics and really don't see any obvious signs of any kind of structural variation across this example of chromosome 12. But some very clever people at the Broad have developed algorithms where we can actually use phasing information, and here using statistical phasing, to improve the sensitivity for picking up mosaic copy number and structural variation. And so here, if we include phasing information and just plot one of the parental haplotypes, what you can see, hopefully, is that we get a deviation away from neutral allele frequency. And occasionally we get a sign shift where over here everything is low, then it switches to high, then low again, then high again. And this is actually demonstrating those phase switch errors that I had mentioned earlier, which are often a consequence of statistical phasing. And so here we can really clearly see that deviation away from neutral allele frequency, indicating that most likely here there's likely a copy neutral or a slight increase in the LR, so maybe a copy gain on chromosome 12 that's resulting in that shift away from neutral. And so we applied this to many of the telangiectasia that were still available from that initial study where we did not identify a point mutation. And in fact, we find that many of them do indeed have some kind of structural variant overlapping the causal gene, indicating that most likely, though there's no point mutation, there could be copy neutral loss of heterozygosity where the germline inherited mutant allele is copied over onto the wild type allele, again, resulting in full loss of function in that protein. And so in conclusion, what we find is that these vascular malformations in HHT and many other diseases that we've studied to this point are indeed caused by biallelic loss of function mutations, whether those be point mutations or structural variations that are a little harder to detect. So I think I'm a little bit ahead of time, but I am finished. So if there's any questions, I would be happy to take them.
Great. Can you use the mic right by you? Thank you so much.
Oh, so official. Hi, I'm Dev. I'm new here. Great presentation. And your work is remarkable. I mean, HHT is a very debilitating disease clinically. I've seen it. It's really bad. My question is, in the 9 individuals that were shown to have the 2 hit, or rather the 7 that were confirmed to be trans, was there a difference in expressivity compared to those who weren't? Assuming would they have maybe had a worse disease progression?
Right. Yeah, so ultimately the sample size is really too low to tell and see any realistic differences in severity. The expectation is that, you might think that, okay, these are the malformations that have a variant, and so maybe would have a worse case. But what we expect is that there are even more types of variants that we haven't assayed and could be present in those samples that we didn't find a mutation. So you could very easily conceive of things like promoter methylation and epigenetics having a role in inactivating the functional copy of the gene, and we basically have no sensitivity for those. So to answer your question, no, we don't see differences in expressivity. We really don't have the power to do that. But also the expectation is that the mutations are there even if we don't see them.
Thank you.
Hi. I was wondering if anyone has ever modeled somaticism. Is that possible to model just like 20% loss of a mutation in a cell and show like a functional outcome or difference?
Yes, so there's been a lot of efforts to model mosaicism. And the one that I'll mention in relation to this work is there's some really beautiful work developing. They're called telangiectasias on a chip, where you can take endothelial cells and put them on a microfluidic chip, introducing kind of flow as normal endothelial cells would expect, and introduce cells with the somatic mutation at any ratio that you would like, and kind of model how that artificial vessel develops. And they do see that you get malformation of the vessel, even with very small inclusions of mutant endothelial cells.
I have time for a couple more. All right, I'm going to give you a fun one. I feel like when I ever go back and revisit the basics, I learned something new, and you did a deep dive into some linkage history. I guess I'm wondering if you learned anything new when you dove back into that, or if there are any fun facts or historic fun facts that you didn't share.
Yeah, I mean, I had a really great time putting together this presentation, reading a lot. I had learned about a lot of the concepts in human genetics classes, but I didn't really have an appreciation of the history of how it came to be. So I had a great time reading through old papers and old perspectives on the history of genetics and learned a lot of cool things, like what I had mentioned at the talk, where I didn't know about knobs on maize chromosomes and how that had been used to actually observe linkage, and many other things that are kind of just interesting bits of history, more so than really fundamental genetics knowledge that remains important today. I mean, I'm trying to think back and think if there's any like nuggets of wisdom, but nothing's immediately coming to me.
Oh, go ahead.
So on the 1984 cytogenetics exam for clinical cytogenetics, there was a question on the first page that asked about what twin spots were. And I worked for a department that did a lot of twin studies. It wasn't that necessarily, but I'm just going to ask you, what do you think the answer is? And I know what the answer is now. So
Yeah, put me on the spot. I actually have no idea what twin spots are.
I was
Sad to say.
devastated. I mean, I was a postdoc at that point, and that was the first time I could take the exam, and I had no idea of what this thing was. And it was like on the first page of the exam. Barbara McClintock actually looking at recombination in maize. I don't think it ever appeared again on the exam.
Wow.
That's a good one. If anyone does nerdy trivia, remember that.
I was actually struck by a table that you showed from the 1996 paper from Neil Reich and Kathleen Mary Congas. Do you mind going back to that? Because I was looking at the ends of the associations. And so if I'm reading that right, is that like the required minimal N of people, right? So they're saying for a variant of a risk ratio of 1.5, that's somewhat common, or 0.02% in the population, you'd need 20,000 people to see a association. Is that right? Because I feel like it's a lot more.
So I think that some of this comes down to the way that they modeled association studies in the paper. And they had been using, if I think back, a particular model of an association study that had been previously proposed on the basis of linkage disequilibrium. And my knowledge of that paper is not really deep enough to be able to say how reasonable that is compared to reality.
Yeah.
So it doesn't surprise me that it is off from what we actually observe doing them today.
It is just interesting to think about it from that point of view and think, oh, they thought we only needed 20,000 people to identify, SNPs for common variants. But, yeah.
But at the end of the day, I think the point holds true that you would need many families to do the same discovery from a linkage-based approach.
Okay. So it was really about comparing the 5 million to the 20,000 and saying GWAS is the better way to go. That was the take home.
Yeah.
I think we'll do this one quick one online. Do you know if people are still doing classical linkage analyses for monogenic diseases? And if so, are GWAS SNP genotyping arrays most useful or superior to exome data?
Oh yeah, no, linkage studies are definitely still being done. And I think they're still very effective for high penetrance monogenic diseases. I mean, the fact of the matter is that for rare disease, you're very unlikely to find any affected individuals within a population. And so, even if you do, it's probably not likely that those individuals are related by, that variant arose in those individuals via a relationship, but rather that those variants occurred independently. And so you can't really use GWAS for doing the same kind of monogenic linkage analysis that you would use for doing rare disease today. So yes, it's definitely still relevant and being used.
Great. I think that is time. Let's thank Dan. Thank you. See you next week.